Hello, everyone, and welcome to today's webinar, Role of Chromosomal Microarray in the Era of Genome Sequencing. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Agilent. To learn more about our sponsor, please visit their website at agilent.com. We encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I would like to now welcome our speaker, Dr. Richard Choy, Professor, Department of Obstetrics and Gynaecology, Deputy Director of Prenatal Diagnosis Center, and Director of the Pre-implantation Genetic Diagnosis Laboratory at the Chinese University of Hong Kong. Dr. Choy will be presenting data obtained using a custom CGH plus SNP array named Fetal Chip. For more information about our presenter, please visit the Biography tab at the top right of your screen. Dr. Choy, welcome, sir. You may now begin your presentation.
Thank you, Alex, for the introductions. First of all, I would like to thank Agilent's invitations. So today, for the next 45 minutes, I'm going to share with you in three aspects. First of all, I would like to introduce about our Chinese University of Hong Kong, and then I'm going to share our experience why there's a need for us to design this fetal chip regarding the principle of CMA, as well as advantage and limitations. And the last part of my talk, I'm going to share with you some of our research findings regarding how we can make use of the chromosomal microarray, and also what's the benefits regarding the need to detect AOH as well as UPD using the SNP array. Okay. So welcome to our beautiful campus. This is our Chinese University campus in Hong Kong. Our campus actually can have many unique features, right? And I extend our most warm welcome to you when you have time to come to Hong Kong. Chinese University Hong Kong is ranked 30th in Asia, and also within the top 50 university under the QS score. And you can see from the red dot here, actually Hong Kong is quite a tiny place, but you can see that we are located within the Great Bay areas in Asia. So that's why we are able to get connected to a lot of other places in the world. Regarding our department, the Department of Obstetrics and Gynaecology. We are the largest public ONG unit in Hong Kong. We are serving about 1.7 million people live in Hong Kong, and we oversee over 7,000 births annually. We are specialized not just in assisted reproductive medicine. We also have UK Royal College accredited fellow training center in fetal maternal medicine, gynecology, as well as other subspecialties. And for the lab that I'm working and take care, actually we are divided into four specialties. We have our endology lab, which mainly, are looking into the test for male infertility. We have our pre-implantation genetic screening and testing facility, which in conjugation with our prenatal genetic diagnosis lab in both molecular and karyotyping, as well as the conventional biochemical screening lab for the down screening. We are collaborating and serving both the reproductive medicine as well as the fetal medicine unit in Hong Kong, as well as in the regions. So we provide both service under our public hospital system, as well as receiving samples from private hospital, including international samples from Thailand, Malaysia, and Singapore. Just like to share with you a little bit about the history of our development. Everything we start from the first trimester down screening in our university as early as in 2003. And subsequently, this down screening program was funded under the hospital authority. So it means that all the public patients in Hong Kong, they were able to receive this as a free of charge service. And because of the screening, actually we understand there's need to develop the cytogenetics as well as a molecular genetic test like the karyotyping and qRT-PCR to provide a rapid and accurate diagnostic test. So all this happens in 2006, and because of the the introductions about the copy-number variants, we start to look into the applications of the chromosomal microarray in the 2004, and subsequently we start to design our customer array and implement it in the surface in 2009 with the CGX SNP. And indeed, we also study and explore its applications in pre-implantation genetic testing for antibody as well. So with this array of tests, you can see this is our hospital campus where we are, and we also have developed another wing of the research unit in China, so that we are able to cater the needs both in Hong Kong as well as those from China as well. Okay, so with this development, the other very important landmark we have done is we collaborate with Baylor College of Medicine, where we start to offer short course and training course in clinical genetics in Asia Pacific region to receive delegates in this region, because we know that's very important for us to learn how to interpret and curate those pathogenic copy-number variants we identified, as well as those of single nucleotide variants. Nowadays we can appreciate by both the exon sequencing or by the genome sequencing. So that's all about our Chinese unit of Hong Kong. So to move on, why we need to do to have a customer-derived fetal chip, and how the CMA is important on prenatal diagnosis. Actually, in fact, all this began in the year of 2004, where Professor Charles Lee and the other group, they published two papers in Science and Nature Genetics to appreciate that actually within our genome there are DNA segments which is present in a variable copy number, like what I show you in the chromosomes, where you see the yellow region is a region where it could get duplicated in our genome. And from that time we start to appreciate that indeed our genome is not as simple as what we know in the old days, right? Our two presidents from America, that we only have SNP difference that's contribute to about 0.1%. In fact, this difference within our individuals, they are not just because of the SNP. There are many small segments of copy number variant changes, like what I show you in this figure, right? No matter we are talking about chromosome 1 or to the sex chromosome, there are a lot of small segments of the changes. And these changes, even in the old days, we know that it could be more than 500 million base pair. And you can see that what I capture in the box is the data from the DGV, which is the database of genomic variants. I'm sure most of you that work in genetics will be familiar with that. You can see that the data in 2010 is only a total entry of about 100,000. And you can see that up to last year, right? You can see that the number of CMEs is almost, increased in 5-fold. It's mainly because we are able to see there are more high resolution data, as well as there are more samples input from the others. So you can see that with this discovery of copy number variants, we now start to appreciate that actually the scale of our genome variant is not as simple as in the old days. We're talking about single nucleotide variant, like the Luna syndrome, that's due to the mutation in the reproductive pathway, that may be a few database, a few base pair, or by indel, to extend that actually there are copy number variant changes. They could be as small as only talking about 50 base pair to as big as tens of millions of base pair. And one of the most representative one is the 22q11.2, which is a DiGeorge syndrome. It could be a 3 megabase or a 1.5 megabase deletions, and that have not including those of other complex chromosomal rearrangement that we can appreciate by karyotyping, or we can also detect it by array if there's a cryptic deletion or duplication. And in the old days, in our prenatal or even in the pediatric, when we're talking about genetic disorder related with birth defect, we always only reference to particularly chromosome numerical changes. That's the aneuploidy. The most representative is Down syndrome. You can see that from this diagram. Now we have a big array of different of disease that we need to take care. And because of this need, when we categorize them into the type of methods that we can detect, we can see that we have been practiced since in the 1970s that any chromosomal disorder, we can use ask our skillful cytogeneticists do cell culture, and then we can identify as tiny as what I show you in the red circuits, that there are 3 copies of the chromosome 21 that's representing one of the most common trisomy. But now that's not enough, because we know that there's a new group of copy-number variant disorder. They could be like what I highlight in the box. It could be a deletion of a small region, or it could be a duplication of a small region, like there's duplication of the blue C square. And this, we need a different method, which is the microarray. Or in the old days, we know that all the Mendelian disorder, they are mainly because of a monogenic disorder that we can characterize by Sanger sequencing. So with that, actually, what we now look for is that can we have a technology that at least can take care of both the chromosomal disorder that I show here, as well as the pathogenic copy-number variant that have been, highly appreciated in the last 15 years with the discovery of new microdeletion and microduplication syndrome. So instantly, what we can think or what we can learn from our lab or from our textbook is that there's a method called the Chromosomal Microarray Analysis. In short, we call it CMA nowadays. So CMA actually, it could be divided into two platforms or method. One is array CGX, which is a comparative genomic hybridizations, mainly looking into region of gain or losses. Or the second one is a single nucleotide polymorphism SNP array that are developed by a different company. But in general, actually, they share a very similar principle. So I'm going to walk through the principle for some of you that are not familiar with this technology. So generally, when we're talking about a microarray, we are referring to that we are picking, clones or segments of sequence our genome, and then we immobilize them, right? These clones into a solid phase. Usually, it's a glass line. So after we immobilize into the glass line, we labeled a genomic DNA that we're going to test in one fluorescent color. Here, I give you an example as a green fluorescence. And then we also have the same amount of DNA from a reference normal healthy control genome that we labeled with a red fluorescent color. So what we do is very simple. Once they are labeled, they are hybridized to the same clone in the array, because if the amount of a sequence is the same and they are specific, they were going to hybridize and give a very unique fluorescent signal. And by looking into these signals, actually we were able to detect the result. Okay, so to reflect this in a more simple cartoon. Okay, so the test samples we label as a red color, and then we have the control sample, they are labeled with a green color. In theory, if both of the genome, they carry the same genomic content, they will be the same. So we will have equal amount of fluorescent signal. But if our test samples have a gain of a particular chromosome or a particular regions, after we do the hybridization, then we have a gain of the red color. So that's how we see there are more red signals. Okay, so to reflect that on a microarray, this is what the microarray scans. Okay, so when we zoom in each of the clones, you can see that by reading out this fluorescent signal, because we know exactly where these probes are. So by reading the signal, we are able can have them aligned back to our genome. Okay, so if there's no signal gain or loss, like what we represent here as a zero line, so you can see that it's a balance. But if we have a gain of the signal from our patient, like in this case, it's a red fluorescent color. So you can see that we see, many red dots, they go to the +1. So we know that there's a gain of copy. But also contradict in the other way, if there's a loss of our patient signal, then that means the control reference signal will increase. So that's why we can see this increase in green fluorescence. So it represents a loss of the signal from the patient. So by doing that, we were able to see and know exactly where there is a genomic copy-number changes in our genome. So with that, actually, it's very robust. We can use different chromosomal microarray platform like the Agilent, the Illumina, or the Alpha Matrix. But now there's only the Agilent and the Alpha Matrix platform. They also have both oligo and the SNP array. So I'm going to tell you why they are better later. But I think for you, the most important thing is like this table I show you, right? In the market, there are many different microarray platforms, and most of the time they will tell you how many, array, how many markers they will have it. But what we need to bear in mind is more important to look at what I show you in the most right is the probe per SNP marker or per marker. That means if they only sample in the same place many times, they still only have the same resolution. They does not enhance the resolution, but the more the number of the probes they have. Okay, so that is very important information. We need to know the resolutions of the array, then we know how good they were able to detect what's the smaller size of the copy number variance they can do. I'm going to explain this a little bit more later. Okay, so to make it simple, the most important key message for microarray or CMA is that it's equal to a high resolution karyotyping. What does it mean? Right? It means that nowadays we know that the best resolution for karyotyping, we are usually referred to a resolution of 5 million base pair or more. That means karyotyping cannot detect any deletion or duplication that they are smaller than 5 million base pair. So just like here, we have a picture, we show the pixel, we know that this is a human individual, but it's hard to tell, who is he or she. It's because of the lack of the resolutions. But by microarray, because we have enhanced the resolutions from 5 million base pair to 100 kilobase pair. So that is a 50 times enhancement in resolution. So with that, what we can see is that we are able to see clearly where the sequence were aberrant. Okay, just like a picture, we can have a very clear resolution, and we know that, oh, this is a gentleman. And in fact, he's the chairman of our department, Professor T. Weiler. So that is what we mean, how a CMA have a better resolution than karyotyping. Okay. So what I'm going to share with you is, okay, what we know about this technology. Okay, why we need this in genetic diagnosis, particularly in prenatal diagnosis, and why we need to move from a catalog array to a customized array that we use in clinical. Okay. So first of all, we all understand that when we're talking about prenatal diagnosis, in the old day, everything starts from down screening. Okay, so we talk about, we need to screen for common trisomy, like trisomy 21, 18, and 13. Okay, because they are the most prevalent. And trisomy 13 and 18, they are even, not comparable with life. But now we all know that this has been taken care well by the non-invasive prenatal testing because it gives a very good sensitivity and specificity, particularly for trisomy 21. But in fact, when we're talking about prenatal diagnosis for high-risk pregnancy, we are talking about fetus with different type of ultrasound abnormalities. It could be as common as high nuchal translucency, like highlighted here in the fetus, that you can see, an excess during the first trimester, or they are simply just by high risk by Down syndrome based serum screening, or it could be more complicated that we see this in the second trimester morphology scan, like this case with cufflet and pilot. Okay, and we were able to get a very precious samples either by amniocentesis, by CVS, or even by chondrocentesis. So with that, there's a need that we need to diagnose lots of different types of disorder, not just talking about common numerical disorder. That's why microarray were able to help to pick up the pathogenic copy number variant. The second reason for need for microarray is that it's not uncommon that we can receive a cytogenetic report where it shows you that this chromosome 7 where the right-hand side one is shorter than the left-hand side, right? And we suspect there may be loss of some of the genetic content of this region, right? So we want to know where is this exact deletion, and also does it affect other chromosomes? And if we know the gene content of this region, we were also able to help the clinical genetic and the genetic counseling regarding what is the clinical significance of this. So with all this drives the need of how we can use the chromosomal microarray. Okay. So we learned all this in the early days in the 2004 and 6, because that is really the blooming of the copyloma variant and microarray. So we feel very excited from that time. Okay, so from that, what we have in the market is like what you can see now, right? We can have iPhone 12, iPhone 5, iPhone 6. The difference, what we know is, of course, the resolution, right? So we take the Agilent microarray as an example. We can have the 1 million probe high density Agilent array. We can also have the low density 60K microarray, but if we are only on 60K, it's called 8 times 60K, is because per array we can do 8 samples. So for the clinical need, what is our trade-off, right? We can go for a Ferrari, that's 1 million, or we can go to a more reasonable local car or a Japanese car that serve the same purpose, but is much cheaper on the road, right? And we know that the main difference is that the higher the density, the oligo or the steep slope, the better the resolution. So with that in mind, okay, it's also the time where the American Congress of Obstetrics and Gynaecology in 2009, they start to issue their committee opinion regarding the use of microarray. They say that conventional karyotyping remain the principal cytogenetic tools in prenatal diagnosis. And array CGH in concert with the genetic counseling can be offered as an adjunct tool in prenatal case with abnormal anatomical finding. So that means even within our professional community, we can appreciate that this new technology has its advantage that is useful, right? But it's not replacing the first tier conventional cytogenetic karyotyping. And why is that? What is the issue? Okay, so I'm sharing with you this is really the first case we do at that time. You can see that at that time, even Agilent, the best resolution is a 244K array. Okay, and in fact, this is a typical first trimester case of trisomy 21, right? You can see that I highlight in the blue box. You can see in the chromosome 21, there's increase of signal, where you can see it's highlighted by the red bar. But at the same time, actually, you can see that across the genome, from chromosome 1 all the way to chromosome 22, we also detect and see there are regions of in the left represent copy number loss, or to the right, which represent a copy number gain. Okay, so for researcher, it will be very excited because you can see there's a lot of copy number variant and you want to know what they are. But if you are a clinician, you just want to make a diagnosis, you will become very worried because in the conventional way by the karyotyping, this is just a typical trisomy 21 Down syndrome baby. It's very easy to counsel. But now you can see there's a lot of other regions in our genome that they are deleted or duplicated. So what's the clinical significance? How should we tell the parents, right, regarding this pregnancy or even for a pediatric case with Down syndrome? Okay, so this raised a lot of issues, particularly two concerns. Okay, the clinicians, they are very concerned about the genotype-phenotype correlation. And for the laboratory director, because you are going to write the report, to easily report to the clinician and the patient, we all worry about the detections of copyloma variants of unknown significance. Okay, and this is not just on one array design. Okay, you can see this is another. This is an ISCA array, and that time that's one of the most popular used clinically. You can see that actually this is on a CAP, quality QAP result. You can see that in this sample, although we can detect there are deletion and duplication on chromosome 8, but again, there are many variants of unknown significance. There's a total of 21. So it makes the interpretations very difficult for routine diagnostic lab. So with all these issues, what we think is that, wow, can we actually remove this known, population-based benign copyloma variants and also some, uncertain segment regions. Okay, just like you can see here. Okay, each dot represents one probe sequence in our genome, and you can see that those in green this is a region that have been known and curated by many groups, like so on the HG18 CMA track there. It's a common copyloma variant, but it's not clinical significant. So what we do is that at the middle one, we try to put in less probes in this region and give a more even space, so that when we do it next time, because by array, you need to have a consecutive 3 or more than 3 probes to call a copyloma change. So by that, you can see that now this region become not detectable, and it is, it's normal. So this is what we mean by remove the benign copyloma variant or those with uncertain clinical significance. And you can see that once we remove that, actually we can save a lot of probes for other regions. Okay, so what we do with the other region is that we try to maintain a high resolution across the genome, approximately 100 kb, but then we try to take out those of benign copyloma variant. So with that, you can see that when we compare our result again with the CUHK array, that is the one in the orange color with 2 cursor, plus the left one, that's our design. You can see that we have the same capacity of detecting the pathogenic one, but we do not see 2 common copyloma variants in this chromosome 14. Okay, so this is one approach. The second approach is that we know that all technology has its limitation, same as on microarray. You can see that although this is a typical example of trisomy 21, you can see that all the signal, they go to the +1. But if you look carefully, for those that are circle, right, they are not respond. They do not generate a very good hybridization signal to give you a trisomy 21. So what we do, we also take this probe out. Okay, so that means we have quite a good and significant, more than a few thousands of probes that we can use to enhance. So what we do with that, actually, we use them, right, to enhance those we know well-defined pathogenic region. So for example, like this one, right, the left-hand side is the Agilent 244K catalog array. You can see that for the Prader-Willi and Angelman syndrome region, for this UBE3A, they are not highly densely populated. So what we do, we enhance the probes in this region so that we were able to ensure that we give a better detection of the region. Okay, so after we finished this modification, this is an example to show how good is this customer design. You can see that this is a well-known region related with Charcot-Marie tube type 1A disease. Is on the PMP22 genes, 22 genes that are shown here. The blue box is the array from the catalog from Agilent. You can see that each of the line represent a probe here. So you can see that from the top, the 1 million density array, they give a very good distribution detection in the region. But if you go down to the 2 times 400K and 4 times 180K, you can see that the probe density in this disease region is very poor. And for the SNP array, actually, they are doing pretty well, but you can see that they are not evenly distributed. It's mainly because they are based on the presence of the SNP in the region. But for our fetal tube, although it's only 60K, but you can see that each of the black color bar here, you can see that we give a much more even distribution and enhance the probe in this disease region. So this is the principle how we design our fetal tube. Okay, so with this design, we also implement with the SNP array, okay, across the whole genome of the chromosome, so that we understand that will enhance us to detect for uniparental, so disomy and others. So that is the what we call fetal DNA chips in Hong Kong and in Asia that we have been using. So how we appreciate that the microarray is a very good approach in prenatal diagnosis. I want to share with you this one of our earlier cases that we understand the beauty of customer array design and how that can help in the patient. Okay, so this is a very typical case that you may encounter in your daily practice, right? This is a referral from a private doctor with suspected cystic hygroma. And at gestational week 13, our ultrasound revealed that it's a high glucose translucency at 9.1 mm with the edema of the fetal scalp. So because it's the first trimester, so we quickly, consent the patient, and then we do CVS. We send for qRT-PCR and karyotyping. And within a day, the qRT-PCR result come back, and it revealed that it's not, we have excluded for common trisomy because high NT could be, highly associated with common trisomy. And it also revealed that actually this is a male fetus. But when we follow up on a morphology scan at week 13, we can see that it is an ambiguous genitalia. We can see that on the left is where you can see the fetus genita, where you can see the right cursor. You can see that this is a tulip sign, right, like the flower tulip. So this is more represent a female genita. Okay, but our qRT-PCR result is male. So actually there's a discordance of results. So this could be a lab error, or it could be this fetus have a hypospatus. But at the meantime, you can see that the nuchal flow is persistently high. Okay, so with that, okay, we wait for another week because the gold standard is wait for karyotyping. And you can see that karyotyping result come back. It's quite a pretty standard and good resolution karyotype. And you can see that it confirms our result. This is a fetus, a male fetus, but morphology scan was a ambiguous genitalia. Okay, so with that, we encounter a very important issue. Okay, normal QF and normal karyotype result, it cannot explain the increased nuchal translucency as well as the ambiguous genitalia. But now we have the microarray. Okay, so what we do, we take the CVS and compare with a normal DNA, and then we use our designed fetal chip to do the analysis. Okay, so within a week, what we see is that you can see that, okay, this is the middle is the zero line. Each of the claws means the plot. You can see that there is a enhanced cluster of plot showing a loss of one copy of signal, which is located in the CYP2 gene. This is only about 200 per kilobase pair. It's too small. So that explains why it was not able to be picked up by karyotyping. But at the meantime, actually, this is the first case in prenatal, see a CYP2. It never been detected. So we look into literature, we know that for CYP2, actually it is known to be reported with Mowat-Wilson syndrome. Okay, and on Mowat-Wilson syndrome, we know that they have some typical features, right? Uplift ear loop, triangular shape, hypothyroidism. Okay, and of course there are some ultrasound abnormality with congenital heart or angiogenesis of corpus. So with all these clinical findings, we were very confident with our rate, and we informed the patient, and then patient go for termination of pregnancy. And you can see that we can see triangular gene, uplift ear loop, angiogenesis of corpus, and hypospadia. And this confirmed as the first case of Mowat syndrome. And in fact, it's not that rare because in the old days they only diagnosed it in the pediatric case because there's a lot of tools to do that. So you can see that our first paper was published in 2010 in Green Journal, but subsequently in 2014 we also published another one because it's almost like every year we do see a case of a Mowat-Wilson syndrome by the microarray. So that explains how a good customer design microarray is important to enhance for the disease detections. Okay, so this echoes all our world leaders in this field like Professor Abudat and David Leatherberg and Lisa Schaffer, that no matter in prenatal or postnatal testing, microarray are taking over the field of cytogenetics for studying chromosomal abnormalities, particularly related with copy number variant. This technology has proven to be useful in several clinical settings, including in both postnatal diagnosis, and now we can see there are great potential in prenatal diagnosis. That was exactly what we see at that time. Okay, and you can see that a few of the landmark publications. For example, this is the one in American Journal of Human Genetics. Okay, it showed that in the pediatric cohort, it gives a higher diagnostic yield, okay, from 15 to 20% for unexplained developmental delay, intellectual disability, and autism than karyotyping. Okay, and in the prenatal cohort, it also depends on the indications of or the estimate of your cohort. It ranges from 1 to 5% or even higher if you have those with more of ultrasound abnormalities. Okay, so with that, actually, it brings up a very important point. It's because, okay, this is a work that we collaborate with Dr. Francesca Gratti. Okay, you can see that for common trisomy, okay, they are related with advanced maternal age. But you can see that for microdeletion syndrome, particularly for the 21 Q DeGeorge as well as the Prader-Willi and Angelman, actually it is not related with the age. So that means any woman, if they go for a prenatal diagnosis, right, the only way you want to detect for this microdeletion and microduplication is you have to go for microarray because karyotyping will not able to detect them. Okay. So with that, actually, it brings up, okay, who should be used, what should be used for daily clinical practice. So to share our experience in Chinese University Hong Kong, we think that one of the most common scenario you may encounter and use is like what I showed you in the example. It's not uncommon you see a high NT cases, but no karyotyping. So this group of patients, they will be very best to use because the woman will always ask, how abnormal is my baby? Is there anything we can take care? Of course, copulomole variant is not the only one. They could also due to lunar syndrome respiratory pathway. But you can see that based on our publication and others, for any fetus with high NT, if there are also other ultrasound abnormality, you are able to have a very good diagnostic yield up to 20%. If they only have NT alone, right? Now we use a cutoff of 3.5, but the first study was only at 3 millimeter. Still, you can have about a 1 to 5% of diagnostic yield. Okay, so in order to have a better idea whether what is the diagnostic yield on different indication, like in the cufflet and palate, we can see that it can go up to about 14%. My student try to do a summary of our result as well as a meta-analysis. So what we do is that we look at the characteristic of this pathogenic copulomole variant. We get all the published data using microarray in the literature from those from the high-risk group as well as those reported in the low-risk group. In a total, we got about 23,000 cases. So what we can see is that actually pathogenic copulomole were underappreciated in low-risk pregnancy. What it means? We know that for high-risk pregnancy, where we refer to is abnormal ultrasound finding, or they have a high risk by down screening, is about 1 in 36. That means 1 in 36 pregnant women, they already undergo an invasive procedure. If you go test for microarray, they will have a positive pathogenic copulomole result. And here we're only talking about those pathogenic copulomole variant. They are less than 10 megabase. How about for the low risk? Right, low risk we refer to that only because of advanced maternal age or because of maternal anxiety. You can see that still is very high. It's 1 in 125. And that is exactly echo the curve I just showed you, the collaboration that we work with Dr. Francesca Gratti, that even for the low risk case, because of the microdeletion syndrome, they are not age dependent. Only microarray were able to help you to detect that. And if we look into more detail, actually you can see that within this 419 pathogenic copulomole variants, most of them, you can see that 84% of them, actually, they are less than 5 billion base pair. So you can say very sure they will be missed by karyotyping. Okay, and we look into more details by the indication. You can see that, right, the highest diagnostic yield is still those with abnormal ultrasounds, and those only with advanced maternal age is low, but still is significant. Okay, and overall, you can see that anyone, if they already go under an invasive procedure, the diagnostic yield of pathogenic copulomole variant less than 10 megabase by microarray is 1.6%. So it actually is echo what Ron Ratner they published in New England Journal of Medicine, that microarray should be recommended as a first tier test. Okay, so that's why we need to choose a good resolution array, because by looking in this pathogenic copulomole variant, you can see that most of them only have a medium size of around 0.47 million base pair. Okay, so you need a good resolution so you can detect all of them. Of course, every technology has its limitation. So in our case, we all understand that for microarray, because we are using probes, and probes could be unevenly distributed, like the net here, all right? Some of the have a more wider spacing, some may have a less one. So this is one of the limitation of microarray. But as far as we understand that, we can still make full use of that. Okay, so for this part, okay, again, I want to highlight is that different manufacturer, different cathode array can have a different resolution. Like this is another region, that is a SIP tool for enhancement carefully. Okay, you can see that, okay, some of the array, particularly those on the SNP array, they have no probe here. It's totally empty. Okay, so you may miss it because there's no probe. Okay, but for for those customer array, like those from us, the fetal DNA chip, or the from Baylor, we have been enhanced the coverage of the probe here. That's why you're able to give a better result. So with that, that's why now actually, right, all the data from us, from the community, and from different publications has support CMA should be used as a first tier test. In the earlier days, only talking about ultrasound abnormality, but now you can see that actually it's recommended for any woman who undergo invasive procedure, right? And they have an advantage also on the turnaround time. In our center is 7 days compared to karyotyping, that's 2 weeks. Yeah. So with all this support, actually in Hong Kong locally, since June 2015, this is also become a publicly available free service for all the women in Hong Kong. And for urgent case, actually, we were able to achieve a turnaround time of 2 days, particularly for those patients, they're already in week 22 or 23, that you need to make a decision before week 24. So we're able to achieve in a clinical diagnostic way, but with the NATA accreditation, and also with a very good turnaround time. Okay, so this is all about the sharing about how microarray were useful. So in the last 10 minutes, I'm going to share with you a study that to show how important to counsel a high-risk pregnancy patient, whether they should use a non-invasive approach or use microarray for the application, and also a few slides, to recap about the importance of AOH. All right. So as like my topic, my title has mentioned, right? We are now in the era of genome sequencing. We know that there's a lot of new technology like, high-def whole genome sequencing, or the low-pass whole genome sequencing like the one developed in our lab. We know that by using low-coverage genome sequencing, we are able to have a better throughput than our rate for copy number variant analysis. But again, for the low-pass sequencing technology, they still need a longer turnaround time. Array, which we're able to give you within 2 days, that is still, clinically very important, particularly for prenatal diagnosis. So with that in mind, okay, I'm going to share with you a very common questions that have been asked by patients. Is that, right, if it's already a high-risk pregnancy, maybe because of advanced maternal age or, other risk calculations, it's common for them to ask, should they go for the NIPT or they should go for the microarray? The disadvantage of microarray is because they are afraid of the miscarriage risk of the extra of 1.2%. But now we all know that, right, it's not that much of this miscarriage risk. Okay, so what we ask the question is that if we have a high risk pregnancy, okay, we have them in parallel test for both NIPS and CMA. Okay, so for the CMA, we use our fetal chips, and then for NIPS, we use genome-wide NIPT at 3 million base pair resolution. This is a study by my PhD student, Ms. Chu here. Okay, so what we group as high risk here is including a patient with advanced maternal age alone, patient with fetal structural abnormality, or they have positive family history. Okay, so and you can see that, okay, in total there are 744 patients, and for the NIPT there are 550 patients have a positive result. That is already excluded the sex chromosome abnormality, and the CMA there's only 32.7% of them have a result. So why there's a difference? First, let's look at the common trisomy. we can see that for the common trisomy, for the red square I highlight, you can see that there's a significant discrepancy in the result, no matter we're talking about trisomy 21 or trisomy 13. One of the reasons is because of the false positive result, but the more important is because of the resolution difference. So let's look at the numerical finding first. Okay, so you can see from here, okay, for all the positive NIPS result related with rare aneuploidy, that means not including chromosome 21, they are all represented by the blue bar. You can see that from chromosome 2 to chromosome 22, we do have more common positive finding in chromosome 7 and 8 and 15, but how much of them were able to have a positive diagnostic result? They are only represented by the orange bar. So you can see that it's very little. So that brings up the fact that there's a lot of false positive results by NIPT compared to a diagnostic test by the microarray. Second, if we look at the size of the pathogenic copyloma variant detected by NIPS and CMA, you can see that NIPS and CMA results were only coordinates in 33% of the cases. Right here, we don't need to mention about the LOH and UPD because this is a unique thing CMA can detect that NIPS cannot detect. But even for the largest one, larger than 5 megabase or smaller than 5 megabase, there's still a high discordance. Okay, so let's take some of them as example. Okay, so within the this 9.8 cases of discordance results, right, we can see that some of the discordance, right, is not just confined to those is larger than 5 megabase. We actually see more that they are less than 5 megabase if you focus on the third column of the table. So what are they? Okay, you can see that one of the main reasons to explain this discordance is that, okay, when we do the CMA, right, this pathogenic finding, apologize if it's too small font size for you. You can see that most of them, they are small pathogenic copyloma variant, right? It's small as 1.6 megabase and 3.5 megabase. So this is one of the typical reasons why NIPT missed it, because of sensitivity of these sequencing platforms were not able to go down to that high resolutions. Okay, and another very important point I want to raise up is that there were pathogenic copyloma variant detected among NIPS negative case. Is as high as 1 in 17 among this high risk group. So that demonstrates the beauty of microarray. Why? It's because some of them are UPD or triploidy that can only detect it by the array. So this is why they missed it. You can see it's about 1.4%. Other is that some of the chromosomal abnormality are missed by NIPS. They are detected by microarray. That may be because of the limited sensitivity and specificity of the platform. And also, if we only consider pathogenic copyloma variant, additional genetic aberration detected by CMA is 1 in 17 among high-risk pregnancy who were given a negative or non-reportable NIPS result. So with that, actually what it shows is that, right, when we compare the application of NIPT versus microarray in prenatal diagnosis among high-risk women, actually, although NIPT can have a high positive rate, but in fact, most of them are false positive. If we only talking about trisomy 21, they're good because we don't miss anything. But CMA able to allow detection of small pathogenic copyloma variants, and they're very important for prenatal diagnosis, right? Just like what I demonstrate here. So for those of you who are interested in this work, you're most welcome to look into the paper from UOG. Okay. Yeah, so this is about all for pathogenic copyloma variant. And as I mentioned, why we also need to have the SNP in our chips design is because we know that uniparental disomy is important. Because uniparental disomy, UPD, means inheritance of both homologous of a chromosome pair from only one parent, or no copy from the second parent in a diploid genome. Okay, so it could be a maternal or paternal origin. And you can see that based on this largest data set from Dr. Thomas from Germany, right? You can see that there are more than 3,600 UPD cases has been reported. And if you look at another publication in American Journal of Human Genetics, where they look into close to a median parents to children pair, you can see that UPD is not just confined to one chromosome. It can go all across all the chromosomes, right? And it could be both paternal and maternal origin, right? So by identifying this information is very important, particularly if there's a possibility of an autosomal recessive condition, right? So to adjust that, actually our customer array, we are also including a enzyme digestion step so that we are able to enhance the detections of the SNP as well. And by optimization, actually, we are able to reduce the volume, and then actually the whole process, the protocol for the testing is, a 30% reduction compared to the total cost. And this uniqueness and the importance of SNP for the detections of pathological UPD or AOX is not just by us. You can see that there are other papers published by the Australian group as well as from the Chinese group that is important to detect this pathogenic insignificant copy-number variants. And in Asia, the other important thing is that we have a lot of consanguinity marriage. Okay, so with this SNP information, actually we are also able to understand, the architect of the family and also look into the consanguinity of the prenatal samples. Okay, so to summarize my talk, okay, this fetal chip is array C6 plus SNP array. It's an 8 times 60K with an average of 100 kilobase genome-wide resolution in the backbone. It has an enhanced probe coverage in disease region. Okay, it has a high sensitivity and specificity in diagnostic yield both in the prenatal and postnatal cohort, as demonstrated in our publications. Okay. You are able to have a high throughput that you can do examples per chip, and it is very robust with a very low experimental failure rate. And as I mentioned, in prenatal, it's particularly important because you have the fastest turnaround time is within 2 days. Right? So far, up to last year, we have conducted more than 15,000 chips. Okay, this including collaborations in Asia as well as in Europe and UK. Okay, so all this as a conclusion for my talk is that actually microarray as the ACMG standard guidelines have been recommended. This microarray analysis is recommended and should be included in both the postnatal and prenatal application. And this is my email. And thank you for listening. And I'm now open for questions. And I pass back the mic to Alisa. Thank you.
Thank you, Dr. Choy, for that informative presentation. We are now going to start the live Q&A portion of the webinar. I would also like to introduce Valentina Moran, the Global Product Manager for Microarrays at Agilent, to join us for this Q&A session. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we're going to answer as many of your questions as we have time for. So let's get started. Dr. Choy, our first question is, what is an average probe spacing in the target sites?
Thank you for the question. So our medium probe spacing for the overall genome is 22 kilobase pair, and in our target region is 5 kb. So that's why, like the first case I illustrate, we have an enhanced probe intensity and how that we were able to detect this kind of small microdeletion syndrome that were missed by other microarray platforms.
Great, thank you so much for that. Moving on to our next question from the audience, are telomeric regions covered?
Oh yes, because the telomeric region is very important, so it's well covered. The mean probe spacing is actually at 2 kb, is highly dense.
Thank you so much. Let's see. Oh, here's a good one for you. So, Dr. Choy, do you see as a risk the massive use of NIPT providing a false sense of security if not linked to appropriate genetic counseling?
Thank you. This is a very important question. Like I show in my last study by my PhD student, you can see that by using NIPT for genome-wide resolution, there's a lot of false positive results. So that brings the importance that actually for this kind of high-risk patients, actually using microarray as a diagnostic test will be much more accurate and appropriate than the NIPT.
Great. And this audience member, number one, wants to compliment you and say very good talk. And their question is, in your opinion, when could WGS replace CMA?
Oh, thank you. I think it's not really replaced. Why I say that is because when we're talking about WGS, which stands for whole genome sequencing, we are talking about a very high red depth technology. Okay, so in theory, with that technology, we can take care of both single nucleotide point mutation that currently take care by West. And we can also, look into copy number variant that were able to be picked up by CMA, or even, balance translocation and structural variant that have been developed in our lab. Okay, so you can see that actually these two technologies, they are taking care of different arrays of disease. Okay, but back if we take the example of in the prenatal, right, it's difficult for each of the case come in and then you do the whole genome sequencing. It's, first of all, it's too expensive. Second, there's too many data and there's so many variants of unknown significance in both copy number variant and single nucleotide variant. Okay, so with that, right, if the primary goal is to make a prenatal diagnosis of known microdeletion, microduplication, and chromosomal abnormality, then there's no way WGS can take over CMA because CMA is cheaper, more robust, and more to a confined detection list. Okay, but in terms of research, of course, there's always a position for WGS to have all this. So I think within a very short, period, within 2 or 3 years, or even up to 5 years, is not ready for the WGS to take over CMA yet in the respect of the confined detection list as well as the turnaround time and the cost of the test.
Thank you so much for that explanation. I do want to remind the audience that any questions that we don't have time for and those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of registration. So let's move on to our next question. This looks like this question is going to be a two-part question. So what are most important regions for LOH in prenatal? And the second part is what is the suggested minimal resolution?
Oh, thank you. So for prenatal, just like, we are following on the ACMG and other guidelines, I think the most important for the AOH or the UPD region is those related with known imprinting gene. So that is the priority where we look at. But the other regions is more like, as I mentioned, is if they are family runs in consanguinity, then we may want to pay attention for those autosomal recessive conditions. They're already known for the family.
Great, thank you so much. And it looks like we do have time for one more question. And Valentina, we have not forgotten about you. This question is actually going to be for you. Is the fetal chip array commercially available? And is it 8 samples per slide, or are there options for 4 or 2 for low throughput labs?
Thank you very much for the question. The array is an 8 sample per array, and it will soon be released within our community design program. So it will be available through the Agilent website. So please, if you have any interest, you can contact your local Agilent representative. We will be more than happy to follow up.
Great. Thank you so much, Valentina. And we want to thank Dr. Choy for his presentation. Do you have any final comments for our audience, Dr. Choy?
Thank you for all. And, I have my email here. So any of you, if you have other questions, you are most welcome to email me or email Valentina, and we will try our best to answer your questions.
Great. Thank you so much. So thank you again, Dr. Choy, and thank you, Valentina, for joining us for that Q&A session. We would like to also thank LabRoots and our sponsor, Agilent, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. And until next time, bye-bye, everyone.
Bye-bye.
Bye.